is Christian Peterson. I'm a fourth-year graduate student in Dr. Woo-Suk Chang's lab in the Department of Biology. I'm here today to talk to you about the effects of a Texas native drought-tolerant inoculant on symbiotic nitrogen fixation and yield potential in different varieties of soybeans. Our model organism is Bradyrhizobium japonicum. It's found free-living in soils across the world, but also forms a symbiosis with leguminous plants such as soybeans. It's applied as an inoculant, also called a biological fertilizer, to soybean fields, and the application has shown to increase vitality and final yield. The main impediment to this is drought, especially here in Texas. It gets really hot, kills the Bradyrhizobium, and then no symbiosis can occur. So we developed a genetic molecular marker system to test intrinsic drought resistance. So then we can take those drought-resistant Bradyrhizobium and apply them as inoculants and ideally increase the yield. So we have one strain specifically, TXVA, that I'm going to be talking to you about today. In figure 1, you can see that Bradyrhizobium species TXVA formed in root nodules on the roots of the soybean plant. Now, when you section those nodules, you can see the red color. Now, interestingly enough, that red color is called leg hemoglobin. It's actually used in the Impossible Burger to imitate the blood color of meat. Just like blood, just like our blood, it does the same thing. It sequesters oxygen, makes a micro-oxic environment so that the oxygen-sensitive nitrogenase enzyme can happen. So what we did in Missouri, the site that I'm going to be talking to you about is a Porterville, Missouri site. What we did in Missouri is we treated our TXVA commercial inoculant, which is called Celtec from Bayer Crop Science, and a non-inoculated control with 3 different soybean cultivars, TN16, which is our Czech that we've used year after year, a drought-tolerant cultivar, S11, and then a drought-sensitive cultivar, S14. Those cultivars were provided to us by Dr. Pingwin Chen from the University of Missouri. In figure 3, you can see the randomized complete block design that we use to minimize any kind of soil variation, and then the drone images of planting and sampling. So what we did was we treated the seeds at planting, then 8 weeks later went back for sampling. Now, why 8 weeks? That's when nitrogenase and the nodules are the most active. So we go back and take random plant samples. At this site, we took 324 samples, and then we looked at the plant vitality. Also pictured here in figure 4 is a drone image from one of our commercial test sites. Down in South Texas, we have larger plots that are more commercial driven. So this is a 30-acre commercial site. And just looking visually, you can tell the difference in the plant vitality. The left side, we have the drought-tolerant inoculant, which is green and fresh looking. And the right side, we have the commercial inoculant, which is kind of yellowed, which indicates lack of nitrogen. So looking at those results, looking at the results of the nodule enumeration, we have taproot and total root counts. With our taproot counts, those are the most important. Because taproot is very indicative of a successful inoculant. Because when you treat the seed and plant the seed, the root emerges. So immediate contact with that Bradyrhizobium on the seed coat will form taproot nodules. Now the total nodules count as well. So you can see some statistical significance there with the total root with TN16, and then with the drought-sensitive cultivar, we have statistical significance with taproot and total root. Looking at our dry plant weight, there's no significance, but we do see a trend that the application of our TXVA inoculant increases the plant biomass. Now, what farmers care about is the final yield. So looking at the final yields, you can see that under every cultivar, the application of our TXVA treatment is improved over the no treatment and Celtec, especially with the drought-sensitive cultivar S14. Now, the conclusions. All of the plants that were inoculated with our TXVA strain showed increased mid-harvest vitality. And then after combine harvesting, the plants inoculated with the TXVA strain showed increased final yield, especially with the drought-sensitive cultivar. This kind of indicates some synergistic effects that we see, and we've seen this across all sites this last year. So we're going to do some more repeated tests at each one of these sites to confirm this. But some more future directions that we have are whole genome sequencing of the TXVA isolate to perform comparative genomics against other members of this family. And then also inserting a green fluorescent protein, the GFP studies, to determine nodule infection studies and get some really, really cool images so we can see the infection patterns and the occupancy of the nodules.